Hi there again. In this video I'll talk about ribosomal RNA gene copy numbers. In the last video I talked about some of the structure of ribosomal RNA genes, but they often, especially in eukaryotes, come in very high copy numbers, and even in bacteria. Normally bacterial genomes have more than one ribosomal gene copy. In this video I'll talk about why that is and why organisms need large numbers of ribosomal RNA genes. And the basic answer to that is all cells need large numbers of ribosomes. As I discussed in a previous video, bacteria have anywhere from about 10,000 ribosomes to 60,000 or more ribosomes per cell, and eukaryotes normally have between 1 million or so, or 2 million, up to 10 or 20 million ribosomes per cell, and some cells have many more times even that number. And this illustrates that a bit. If you look inside of a bacterium, you see around the DNA a large number of ribosomes in each section, and if you count all of those up, you find that a small bacterial cell will have 10,000 or more ribosomes, and larger cells will have many times that number. And then if you go to eukaryotes over here, you can see the large number of ribosomes that are attached to the endoplasmic reticulum, and there are even more ribosomes floating in the cytosol, those cytosolic ribosomes as well. So you have literally millions of ribosomes in each eukaryotic cell. And in order to form or to produce all of those ribosomes, you need the ribosomal RNA and all of the ribosomal proteins that go together to make a functional ribosome. And that presents a challenge for organisms to produce enough ribosomes so that they can make their proteins, including times when they're growing and dividing, when they need to essentially double the number of ribosomes they need as they lead up into cell division. And as I was explaining in the previous video, the ribosomal RNA genes come in multiple copies in eukaryotes, as well as bacteria and archaea, for the most part. And those are then transcribed. Those genes are transcribed, and the RNA is processed, bundled into small subunit and large subunit ribosomes, and then shipped out, transported out of the nucleus through nuclear pores to become functional ribosomes once they attach to an mRNA, and then accomplish translation of the mRNA into proteins. Some of these proteins then that they're forming are ribosomal proteins. Others are proteins that wind up in the cytosol, on membranes, in organelles, etc. So to produce all those proteins in this rather complex system, you have to produce a very, very large number of ribosomes. So then if you look at the numbers, and we already talked about this a little bit, bacteria have between 10,000 and 60,000 ribosomes per cell. Eukaryotes from 1 million to 20 million, and some actually have even more than that. RNA polymerase can synthesize about 50 nucleotides per second. In some bacteria, maximally maybe 100 nucleotides per second. If there is only one ribosomal RNA gene, that is, one RNA gene available, approximately 6 kilobases in length, it would take 6,000 divided by 50 times 10 to the 6 seconds, or 1.2 times 10 to the 8 seconds to produce enough RNA to make 1 million ribosomes, that is, this 10 to the 6th number. Well, that turns out to be 3.8 years. So that's enough to make 1 million ribosomes for one cell, one eukaryotic cell. So to undergo growth and replication and cell division, for a eukaryote to have only one ribosomal copy would take about almost 4 years. And for bacteria, that would take about 14 to 83 days to make 10,000 to 60,000 ribosomes. And to produce 20 million ribosomes in some larger eukaryotic cells would take about 76 years. So something is not quite right here. And obviously this is not occurring. So the first thing that organisms do, and I've already shown this picture previously, is that they don't just transcribe one RNA at a time. In ribosomal RNA genes, the RNA polymerases, polymerase 1 in the case of eukaryotes and RNA polymerase in bacteria and archaea, they pile up those RNA polymerase molecules on the gene and produce multiple transcripts at the same time. So by packing 100 molecules, 100 RNA polymerases on that gene, then you can speed up ribosomal RNA production by about 100 times. But still clearly, more than one ribosomal DNA is needed. One ribosomal RNA gene is needed. So let's look at some data here, actual data from real organisms. If you look at Tetrahymena, which is a ciliated protozoan, a eukaryote, in a haploid nucleus, you only have one ribosomal DNA copy. So it looks like that shouldn't work, and those organisms shouldn't be living, but they are. So I'll show you why it only has one and how it adjusts for that. Mitochondria and plastids have 1 to 3 copies of ribosomal DNA per genome, but that's not quite enough either, so I'll show you how they accommodate for that. Then you get to E. coli, which has 7 copies, and remember, E. coli can, at its fastest, divide every 20 minutes. So are the 7 copies enough for that rate of replication and division? Saccharomyces cerevisiae yeast has 140 copies, so now you're starting to see what these organisms are doing. They're maintaining more copies, including piling up the RNA polymerases. They're piling them up on multiple copies of those transcription units. You have about 200 copies per haploid nucleus, or 400 for your diploid cells. Salamanders have from 5,000 to 10,000, and plants have from 200 up to 22,000 copies of ribosomal DNA. So clearly something's going on here too with these organisms. So here's Tetrahymena, which is a ciliated protozoan, and you can see the cilia all over this cell. And it has, actually, when you go inside and look inside of that cell, which is one of those protozoans that's undergone multiple endosymbiotic events, it has 2 nuclei, a macronucleus and then a micronucleus. And if you look at the micronucleus contains the genetic material primarily, the genetic material that's passed from one mother cell to the daughter cells. The macronucleus is polyploid in that there have been many divisions and replications of the chromosomes, but the nucleus has not divided, so it's polyploid. And so that kind of hints at what's happened here. This one has only one ribosomal RNA gene in its haploid genome, but then it becomes polyploid, so then you're making multiple copies of those ribosomal genes. So right when you have the initiation of the macronucleus, which starts out small and then grows, this is what the locus for the ribosomal DNA or ribosomal RNA looks like. Here in yellow, that's the ribosomal transcription unit, which contains the small subunit gene, the 5.8S gene, and the large subunit gene. But then surrounding this, you have some repetitive sequences, A prime here, a couple copies of that on this end, and A prime on this end, and then M sequences also throughout this, and I'll show you what those are. So the first thing that happens on the initiation of the macronucleus is there are site-specific breaks using enzymes, endonucleases, which cut right between those M and A prime sequences on the 5 prime site of the gene as well as the 3 prime site of the gene. Next, the ends of the main parts of the chromosome are then protected by adding telomeres to the ends of those. So these repetitive sequences are added to the end so that there's no further degradation of those parts of the chromosomes and the parts of this ribosomal fragment that's being chopped out. Then the end without the telomeres and has these M sequences on it, loops around and forms a hook on the end of that DNA fragment. Next, there's strand invasion, and then DNA replication begins in that circular region on the end of that DNA fragment. And replication continues. There's a leading strand and lagging strand here. And now you have 2 copies of the ribosomal genes that are formed on this fragment. And then replication continues all the way to the end. And so then you have a fragment of DNA now with 2 copies of the ribosomal genes. So not only does the genome become polyploid in the macronucleus, but for each of the ribosomal DNA loci, those duplicate. And so those are being chopped out and duplicating. So then you have even a larger number of ribosomal RNA genes or ribosomal DNA. And when you look into the macronucleus of tetrahymena and other ciliated protozoans, which do the same thing, you find these little fragments. They're actually quite large fragments, a couple of tens of kilobases long each containing 2 copies of the ribosomal DNA. So through polyploidization and duplication of the ribosomal genes, then tetrahymena has hundreds of copies of ribosomal DNA from which to make ribosomal RNA and to produce its ribosomes. E. coli has 7 copies of ribosomal DNA, and this is for all of the strains of E. coli, I think, have 7 copies. So how long would it take to make 20,000 ribosomal RNAs for its 20,000 ribosomes, if it only has 20,000? Some have a few more than that. The transcript in this case is 5 kilobases in length. The RNA polymerase, again, will polymerize at about 50 nucleotides, sometimes a little higher than that. Therefore, the polymerization of approximately 5,000 times 20,000 nucleotides are needed, which is about 100 million nucleotides are needed every 20 minutes for it to divide. So at 50 nucleotides per second, it would take 2 million seconds if only 1 ribosomal DNA transcript was being used. Since there are 7 copies, then that would mean approximately 283,000 seconds, or 3.27 days would be required for each E. coli cell to divide. But as with all cells, and this is for an E. coli cell, Again, it lines up its RNA polymerases along the DNA, along the transcript, and transcribes about 100 copies simultaneously and continuously. So by packing these 100 or so RNA polymerases on a single gene copy simultaneously, then now you can divide this 283 by 100 or so. And now you have 2,380 seconds, which is 40 minutes. If you speed up polymerization just a bit, and they've shown that some bacteria can polymerize RNA at a rate close to 100 nucleotides per second, then you get down to about 20 minutes. So with just 7 copies, you can divide every 20 minutes. Now, some bacteria actually divide, it takes them longer to divide. So some, it takes half a day or so. And you find that those ones have fewer copies of ribosomal RNA genes. And so some of the rates of cell division of colony doubling and things like that in bacteria are related to the number of ribosomes that the organism can produce in order to divide, and that's all dependent on the number of ribosomal RNA gene copies that are in the genome. So what about mitochondria and chloroplasts, which have between 1 and sometimes 3 copies of the ribosomal RNA genes within their genomes? What happens with those is that they also become polyploid, similar to how tetrahymena is becoming polyploid to produce more ribosomal DNA copies. So for each mitochondrion, you don't just have a single gene or genome copy in each organelle. You have multiple copies, and in chloroplasts the same. You don't have just one copy of the chloroplast genome per organelle, but you have multiple copies per organelle, and that's probably because they need more ribosomes and more ribosomal RNAs, but probably some of the other genes are needed in higher copy numbers too, such as the genes that are responsible for some of the photosynthetic processes. Here's a view of chloroplasts within a plant cell, stained with a stain called DAPI, which is a fluorescent dye, which fluoresces in the blue for DNA, and red here in the case of the pigments for photosynthesis. But you can see in each chloroplast here, each one of these is a single chloroplast, there are multiple little dots. And each one of those dots is a genome. So these, you can see that they're polyploid. They have many genomes per organelle. And sometimes these are actually joined together like beads on a string. In this case, some of these look like they might be, in that you have this circular appearance of these little beads, of which each one of those is a genome's worth of DNA for each one of those organelles. As I showed you in that table, many eukaryotes just maintain a large number of ribosomal RNA genes within their genome. So plants have about the highest number and range. One plant from broadbean, one of the things that I studied for my PhD, one plant might have 200 copies per genome, 200 ribosomal RNA copies, while others had up to 22,000 copies of ribosomal RNA genes. And actually, if you looked at the two plants, they look the same. So obviously this one has a lot more than it actually needs. And so there's some transcriptional control in this case to not transcribe from all of those, but at least 200 is probably a minimum in this case. So within a single genetic line, there could be as much as a 20-fold difference, and in a single plant, you could have up to a 12-fold difference from one part of the plant to the other, indicating that the number of ribosomal RNA genes in each cell in each plant the numbers are not the same, such that in one cell you could have maybe 200 copies, and in a cell in the same plant you could have over 1,000 copies or even 2,000 copies in that cell. But in an individual cell, then you have these transcription units. Many of them are transcribing at their maximum levels. So you have 100 polymerase molecules here, polymerizing transcripts. There's another one here, here, here. So you have simultaneously usually hundreds of these transcribing all at the same time. And some of this information came from what I did for my PhD, which was to look at ribosomal gene copy numbers in this broadbean, Vishafaba. And I looked in many different tissues, looked throughout the plant, and in a population of plants also. So the project started out this way. I went to lunch with one of the graduate students, senior graduate students in the lab, and she talked to me about, I think, 3 different projects that maybe I could pursue in the lab. And this is the one I chose, and she showed me a picture like this, where DNA had been extracted from Vishafaba. It had been digested with a restriction enzyme and then hybridized to a ribosomal RNA probe, showing the pattern within that RNA locus, that ribosomal RNA gene locus. And there was one band that was strong down here at about 3.8 kilobases, and then there was a whole ladder of bands. And then if you look at the separation on this ladder, they were all approximately 325 base pairs apart. So that's what the project was, find out what's going on there. So I started doing these southern blots on a large number of different individual plants and found that the patterns varied quite a bit from one plant to the next to the next. We mapped this region, and it turned out that there were ECOR1 sites, one at the end of the small subunit gene, or 18S ribosomal RNA gene, one near the end of the 25S, or large subunit gene, and that was it. And so this region here, between those 2 ECOR1 sites, was this 3.8 kilobase region. Whereas this region here, between the end of the 25S and the first part of the 18S, including the intergenic spacer, or non-transcribed spacer, was variable. And it was variable by 325 base pairs, and we tracked that down to having these sub-repetitive elements of 325 base pairs each. And I mentioned those previously, that those are regions that look somewhat like the promoter. And so the RNA polymerase can stack up in those areas, ready to hop on to the regular promoter to start transcription, so that you have plenty of RNA polymerase ready to go in that locus. But in addition to doing those types of blots, southern blots, I also, by a similar method, but one dot per hybridization spot, I measured the amount of ribosomal DNA in several different types of tissues. So individual leaves from individual plants, cotyledons, embryos, roots, etc. And found that there was a range ranging down here. This would be approximately 200 copies of ribosomal DNA to a plant up here that had roughly 22,000 ribosomal RNA genes, which is over a 90-fold range of difference from one plant to another, one leaf to another. And this is a summary of that. So in leaves, the average was 2,500 copies per haploid, which agreed with previously published papers. So that was, but there was a 230 to 22,000 copies of ribosomal DNA per haploid in the 434 leaves that I studied. In the cotyledon, up to a 45-fold variation, embryo 6-fold. And then you go down to this, various tissues in the same plant, up to a 12-fold range, even in an individual plant. And then I did some genetic crosses, and there were also variations there in the number of ribosomal genes. So this pointed to the fact that there were probably differences from cell to cell to cell in every plant that I looked at. So then I did self-pollination in this plant to see from parents, the P here, to progeny, the F1, were the copy numbers being inherited essentially in a Mendelian fashion. So here we have on top the parents and then the progeny, and in some cases you see in 17 here, there's the parent, the number of ribosomal copies, and all the progeny had more than that. And then you go to some other self-pollination experiments, such as 38 down here. Here's the parent here, and all the progeny had fewer ribosomal gene copies. And if you look all through here, it's pretty much random as to whether they had higher amounts, lower amounts, or about the same number of ribosomal copies in the parents and progeny. So already this was pointing to an epigenetic effect, and clearly a non-Mendelian inheritance or non-Mendelian changes in these ribosomal gene copy numbers. And that was at a time when epigenetics had not even been a subject that people were talking about. So these were changes independent of Mendelian type inheritance. In addition to self-pollination, I did some genetic crosses. So I took parents that had some that had about the same gene copy number, crossed those, looked at the F1, and then allowed the F1s to self-pollinate and looked at the F2s. And the overall take-home of this is you could have parents with high numbers, progeny would have lower numbers. Some had the same numbers of copies in the parents and the progeny, and some had lower numbers. So again, a non-Mendelian type of genetic effect. So the copy numbers, ribosomal gene copy numbers, were changing, and they were changing within plants. And that characteristic of having more or fewer or the same number of ribosomal genes was not a Mendelian type character. That is, it was not inherited from parent to progeny. So then if you look at the ribosomal gene locus, and I talked about this slide in a previous video, here's the secondary constriction with all of those ribosomal genes in one place. They are then transcribed into a long transcript, which is then processed and made into and then incorporated into the ribosome. Now, as I said, down here in Visha faba, you have these 325 base pair subrepetitive elements that attach the RNA polymerase, and so they're from anywhere from 0 of those up to 23. And then if you look in other plants, you find the same thing, and if you look into humans, you find the same thing, and if you look in mice and birds and things like that, you find similar things. That is, these subrepetitive elements in these intergenic spacers. And so I looked at these in these southern hybridizations again to see what sorts of things were changing. And as you can see, there are always these ladders of bands, and each one of those bands varies by 325 base pairs. And the subrepetitive elements are also 325 base pairs apart. So there's a connection there. So when you do these crosses, what you get is a blending first of those different types of bands, and each one of those bands represents an IGS with, let's say, one one of the subrepetitive elements, and then 2, and then 3, and then 4, and then 5. And so they vary by the number of those subrepetitive elements that they have, and that's indicated by the lengths of those DNA fragments in the gels. But when you do these genetic crosses, you would expect that they would just be a blending, in this case, between the pattern from parent 1 and parent 8. And essentially they are, but then when you look at the F1s and F2s, and the F2s are the progeny of self-pollinated F1s, so these are self-pollinated, you do get something that looks similar to the type 1 pattern and the type 1 and the type 8 pattern, as well as the heterozygotes. But you also get ones that differ significantly from those. And so this is a first indication that there's some sort of genetic recombination going on. And another part of the information that I generated was I did some R-looping, which is where you mix the ribosomal RNA with the ribosomal DNA, those gene loci. You denature those and let them reanneal, and then look at where the displacement is. So in this case, if you look over here, essentially the ribosomal RNA has displaced the DNA, so then you get a single strand of DNA looping out here. Here's the 18S, where the 18S ribosomal RNA has displaced the DNA, so you have an RNA-DNA hybrid in the middle, and then the little loop out, the thinner strand, is the DNA that's been displaced. So you can follow these through. Here's the 25S, and then you go through here. There's the 18S and the 25S again. There's the 18S and the 25S again in this electron micrograph, and then this is the interpretation down here. So when you look at the intergenic spacers, then they're of different sizes, and this led to part of the conclusions also, which I'll show you next. So what this all indicated was that there was recombination going on, unequal crossing over, within mitotic cells, that is somatic cells. So it can happen during meiosis in the flowers, but it also looks like it's happening in the leaves and the stems and the roots and everything. So if you have these intergenic spacers lined up, they can vary with how they line up according to those subrepetitive elements. And if you get a crossover during that time, then you can generate new versions of those intergenic spacers, varying by the number of those 325 base pair subrepetitive elements, which is where the polymerases are sticking. So those, by that mechanism, you can generate new versions of those 325 base pair repeats, and therefore each one of these IGSs is slightly different, and thus you get that ladder of bands, because this one with 4 is shorter than the one with 7, so the one with 7 will be higher on those gels, and the one with 4 will be lower. And so eventually you have a series of bands representing intergenic spacers with anywhere between 0 of those subrepetitive elements up to 23 of them. But the average number was right around 6 of the subrepetitive elements. On a longer range, you can actually line up those chromosomes differently, so that I've just colored them here. So you can line those up because they still have those 325 base pair repeats in common. And you can get crossovers so that you can change the overall ribosomal gene copy number on each one of those chromosomes. So this is kind of an extreme example, but I've lined these up very differently so that you get a crossover. And the resulting chromosomes look like this, where one has a much larger number of the ribosomal gene copies, and the other chromosome that results from that has fewer copies. And as you get down to a minimum level, 200 copies, then if you get any smaller than that, those cells are non-viable, so they can't make enough ribosomes. But in general, organisms, eukaryotes, use this method, this unequal crossing over within the ribosomal region, to maintain a large number of ribosomal gene copies. And again, this is a non-Mendelian. It's not inheritable. You can't inherit the number of copies. The copies are changing from cell to cell to cell. So this is an epigenetic effect rather than a Mendelian type of mechanism. So I want to mention a few other things. In oocytes of animals, you have very, very large cells. In fact, some of these cells in amphibians you can see with the naked eye. They're huge, huge cells. And they can have up to 10 to the 12th ribosomes. That presents even more challenges. So in amphibian cells, the approximate number of ribosomal copies is about 200 copies per haploid. So clearly, to get 10 to the 12th, 200 copies is not going to do. Therefore, there are about 1,000 copies in a primary oocyte, that is, one at the 4N level, prior to division 1 of meiosis. And each division is asymmetric, so you wind up with only one mature oocyte. But during this time, the number of ribosomal genes is increasing, and the number of ribosomes also is increasing. So by increasing the number of ribosomal gene copies alone on the chromosome, there are some benefits as far as producing more ribosomal RNA and more ribosomes. But these organisms also have the ability to extrachromosomally amplify those ribosomal genes, and that's the key to creating enough ribosomal RNA and ribosomes. But in addition to having more copies of the ribosomal genes and ribosomes in an oocyte, they also have what's called extrachromosomal amplification. So again, as you saw in Tetrahymena, they split off individual ribosomal genes, and they start with about 200 to 1,000. And those small fragments then circularize and start replicating, just like small chromosomes. But they do so in what's called a rolling circle mode of replication, which is similar to how viruses replicate their genomes. And so you have the leading strand going around in one direction, and then this fragment coming out, this piece coming out, is the lagging strand of the replication. But eventually you wind up with these small mini circles that contain ribosomal copies, ranging from about 1 copy up to about 20 copies. And these then produce the huge number of ribosomal RNAs and ribosomes needed in those oocytes. So again, here's the first part, just to accentuate that. Here's the 2N oligonium. It builds up the number of chromosomes, duplicates its chromosomes to a 4N level. But then the first division is asymmetric. That cell is larger. Then there's a second meiotic division where it's also asymmetric. And finally, you have the mature oocyte, which already has a much larger number of ribosomes already and ribosomal RNAs than the original cell had. So it's already built up the number of ribosomal RNAs that it has in ribosomes. Sperm don't do that, right? So you start with a spermatogonium, duplicate the chromosomes, and then make 4 sperm with a much, much smaller number of ribosomes than in an oocyte. And one reason that the oocyte or egg cell needs a much larger number of ribosomes is that these first divisions after fertilization are called cleavage divisions. So the overall mass of that cell and group of cells is not changing to any great extent, but you're dividing up essentially the cytosol of that cell while also duplicating the chromosomes. So you need a large number of ribosomes to make it through that process. And then later, during the blastula and gastrula stages, then the cells are having more of a appropriate level of ribosomes produced in the 2 million to 10 million ribosomes per cell numbers. So that's the end of this material. In this video I tried to explain why organisms need so many ribosomes and how they accomplish making enough ribosomal RNA and ribosomes for each of those cells, ranging anywhere from a low of maybe 10,000 ribosomes per cell up to 10 to the 12th ribosomes in a large oocyte. So several mechanisms have evolved to perform those functions of making sure there's enough ribosomal RNA, including extrachromosomal amplification, unequal crossing over, just maintaining a large number of ribosomal genes. But in essence, it's necessary for the survival of these organisms to have enough ribosomes in order to make all the proteins they need to grow and eventually divide. Okay, so that's it for today. Thanks for watching and listening. I'll talk to you later.